Hello Year 12, welcome to the exam analysis from your mock exam in biology. I'll be going through all of the questions and with some answers and some tips to share with you. I hope you enjoy it. So let's get started. Okay, so looking at the first question, it gives you some information about white blood cells and bacterial cells, and particularly it says complete table 1 by putting a tick in a box if the structure is present in the type of cell. So looking at the answer, what you would include is the following. Basically, white blood cells, eukaryotic cells, they have a cell surface membrane and also there we go, cell surface membrane, and they also have a nucleus. Bacterial cell is a prokaryotic cell, so it does have a cell wall, okay, and it does have a cell surface membrane, but it doesn't have a nucleus, okay. It has DNA, RNA material, but it's not in a nucleus. Question 1b says figure 1 is a diagram of a mitochondrion at a magnification of times 30,000, and it says calculate the actual length of this mitochondrion in micrometers. So it says show your working. So what we've got to do is obviously the actual length. What we've got to do is remember the basic formula. So if you remember, I is the image divided by the actual times magnification. So that's in our equation triangle. Not a very good triangle, but never mind. And if you look, you can work out, well, the actual length is basically the image size divided by the magnification. Okay, so we know the magnification, which is good, which is times 30,000. So that's 30,000. And now we've got to measure the actual length. So you would measure from one end to the other with a ruler, okay, in millimeters. And I make that to be about 82 millimeters, okay, so 82 millimeters. Okay, so I want to convert it into micrometers, so I times it by 1,000. Okay, so times it by 1,000. And so now I can work out the answer, which is basically 82,000 divided by 30,000. In this case, what I get is 2.73, which actually, as I look at the mark scheme, it's incorrect. Oh dear, I've not measured it correctly. I think that therefore is 83 millimeters. Okay, next question. Looking at the theories that mitochondria are organelles that evolve from prokaryotic cells, it says give one piece of evidence that supports the theory. Well, in fact, there are lots of different pieces of evidence, and you could have things such as circular DNA, for example. You could have had the fact that they're smaller, or they've got 70S ribosomes, okay, they've got no introns, no histones, and proteins associated with DNA. And then for 2 marks it asks, what is the advantage to cells of having mitochondria? Well, the first thing is basically they're able to respire aerobically. And I think some of you forgot to use that word aerobically, okay, because that's with oxygen. It's quite important to state that. And then the second question, well, basically, so to make more ATP, or you could have had a release, for example, release of energy. It's important to state here, a lot of people make this mistake, that they say that that the mitochondria produce energy. Okay, that's rejected, right? Because they don't produce energy, they're releasing energy. They're making ATP to release energy. They don't produce energy. Nothing produces energy, as we all know from our physics GCSE. So just be very, very careful with that type of question. Here are the answers for question 1. You might want to pause if you're writing on your test the answer. Okay, this is question 2. I know it says question 3, but this is question 2 in your test. And it's related to enzymes, and it's telling you lipase hydrolyzes triglycerides, and tells you about a student investigating hydrolysis of triglycerides in milk by human lipase at 20 degrees C. Okay, so 20 degrees C. Recorded the pH before and after adding lipase, and you used a pH meter to record pH. Then it says it shows a graph of the results, so let's have a look at the results. Okay, there we go. So you got pH, lipase added. So obviously as the lipase is added, the pH is reducing, it's becoming more acidic, and then it's leveling off for a particular reason. And the first question is really testing your kind of understanding of apparatus and techniques, and also practical skills. And it's saying suggest one advantage of using a pH meter rather than a pH indicator in this experiment. Well, a pH indicator is basically it's a colour change, so it's very subjective. Doesn't give quantitative data, and it's basically not as good as a pH meter, because pH meter can give you a numerical reading. It also can give you greater accuracy as well. If you said greater precision, that would be acceptable as well. Okay, 2b, explain why the pH decreases when the lipase is added to the milk. Well, if you know about hydrolysis of lipids by lipase, you know that several things will be produced, particularly fatty acids. Okay, so now fatty acids obviously are going to have acidic. So you can't say acid, because it's not specific enough. You need to mention fatty acids. And then it says suggest why the pH remained constant after 2 minutes. Again, that's for 2 marks. Okay, so you need to go back to the graph and you need to look at the graph. I'm looking at the graph and I can see after 2 minutes it levels off. And basically, a lot of you again gave the first point that basically all triglycerides are used up, okay, but you didn't then go on to say why. So why? So basically why is because no more fatty acids are produced. Final part of 2D, so it says the student carried out another experiment at 15 degrees C. It says draw a line in the graph in figure 3 to show the results you'd expect. Well, that's at 20, okay, so for 15 it's exactly the same point, but because hydrolysis is going to be slower because it's a reduced temperature, okay, you're going to have, it's going to be above the line, okay, and then that's for 1 mark, and then it's going to actually level off at the same pH, but a bit later, not much later, a bit later. So if your graph did something like that, then you would have got the 2 marks. Okay. The explanation is that basically less fatty acids and glycerol are being produced because it's a slower rate of reaction. However, it's 15 degrees C, so the reaction will occur, but just at a slower rate. Answer to question 2. Again, you may want to pause. Okay, on to question 3 in your test, okay, and this one is about trypsin. So it does tell you there, trypsin is a protease, okay, so it says it produces an inactive form inside some of the cells of the pancreas. And this one says, name the part of a pancreatic cell that produces the inactive form of trypsin. And the first thing your brain looks at is the word pancreatic cell. And you probably go through your data bank of information and you go, but Sir hasn't really told us about pancreatic cells, so I know nothing about pancreases and their cells. Well, that's not what it's looking for. It says name the part. That's the key thing. The part of the cell that produces the inactive form of trypsin? Well, they give you a whopping great big clue in the stem of the question. It says it's a protease. Okay, so protease, it's made of proteins, so therefore the answer must be either ribosome, or you can have the rough endoplasmic reticulum. Okay. Next question says suggest the advantage of producing trypsin in an inactive form. Okay, so if you think about it, you don't really want protease to be digesting proteins inside the cell. So basically, it doesn't digest proteins inside the cell. And basically, for the second one, so that the cell or the tissue or the pancreatic parts of the cell are not destroyed or damaged. And I think some of you again didn't go on to say that. That's all it was looking for in that exam question. 4CI, so again they've given you some information there that we're going to look at in a minute, but it's saying name the type of a bond hydrolyzed when the short chain of amino acids is removed. That is just basic recall. Okay, so you've got to remember that bonds are being hydrolyzed, okay, for proteins, which are basically peptide bonds. Okay, again, some of you are putting different types of bonds in there, but it's a protein, so it has to be a peptide bond. Okay, this question, you got 3 marks here. Okay, so they're looking for 3 main points. So it says sometimes trypsin can be activated inside a pancreatic cell, and it says a competitive inhibitor in the cell then binds to the trypsin and stops it working. Okay, so it's explain how the competitive inhibitor stops trypsin working. Well, the first thing to mention is about the inhibitor itself. So the inhibitor is a similar shape to the substrate, okay, and therefore if it's a similar shape to the substrate, it's going to block the active site. Or you could say it's complementary to the active site or binds to the active site. And that's for the second mark. Then for the third mark, the substrate can't bind to the active site because it can't actually get in there. So I was pleased to see some of you putting that there were no or fewer enzyme substrate complexes formed. Okay, there's the answer. There were a few answers that were a bit disappointing because it does say a competitive inhibitor. Unfortunately, some of you talked about non-competitive inhibitors and how they fix to other parts of the enzyme and change the active site. I think that's just a basic lack of revision there. Just be very, very careful. You know which type of inhibitors do what particular functions. Here's the answers to question 3. Okay, next question is about lungs and breathing, and it talks about the forced expiratory volume as well. First question, it says explain the role of the diaphragm in breathing out, and just to note it is worth 3 marks. So for the first mark, it's looking for the fact that the diaphragm moves up when you breathe out, or it becomes dome-shaped. For the second mark, it's looking for the fact that this will reduce the volume of the thorax, or you can say it increases the pressure in the thorax. For the second mark, they'll also accept space for volume and chest, lungs for thorax. So for the third answer then, it means that the pressure in the thorax becomes higher, and that's a key thing. You must mention the word, it's higher than outside. Okay, so that's for the third mark. They will accept chest or lungs as well for thorax. Okay, and then this next question of 4B, and they give you a lot of information here that you need to spend time going through. Okay, so it tells you about the forced expiratory volume is the greatest volume of air that a person can breathe out in 1 second. It tells you about what scientists did and how they measured. They remeasured each person after 1 year, calculated the mean, and then repeated at the end of the 5 years. It then shows you that the scientist results. Okay, and you need to look at those very, very carefully. So that's the time in years, that's the mean FEV1 per dm cubed, and then people who stopped smoking, people who continued to smoke. Okay, so this question, it says use the data, and in bold it's got figure 4, so you have to use the data to compare the change in FEV1 of people who continued to smoke with those who stopped smoking. And again, there's 2 marks. Okay, so looking at the graph, as you can see, so particularly people who stopped smoking, people who continued to smoke. So the first thing I would point out is this after 1 year. So obviously a difference between the two. Okay, so you can actually see after 1 year, those who've stopped smoking, it increases after 1 year, whereas the FEV1 of smokers decreased. Okay, so people who continued to smoke decreased. So that's only 1 mark. For the second mark, they're looking for basically to mention that between 1 and 5 years, you can see the people who continue to smoke, there's a faster rate of decrease than those who stop smoking. So it's a faster rate of decrease. You must get that in. Okay, you could quote some data as well if you wished. And question 4c, it says smoking causes changes in the lung and airway of smokers, and it says suggest. I love that word suggest, because there's lots of different answers for this. It only wants 2 changes in the lungs of people who continue to smoke. Types of answers that are acceptable are things like airways are narrowed or blocked. There could be excess mucus in the airway, inflammation of the airway, or elasticity is lost, or scar tissue builds up. In terms of the answer, the examiners are told to ignore answers in the context of reduced surface area of alveoli or increased diffusion distance. The reason being is that they want you to explain 2 changes in the lungs. They don't necessarily want you to talk about a wider context of the alveoli, for example. They want you to talk about the lungs. Okay, here's the answer for question 4. Again, I would pause it and update your answers on your exam script. Okay, on to question 5 in your test. Obviously there's a lot of information in here. This type of question is obviously going to test your knowledge of enzymes, but also in particular it's testing your knowledge of how to do practical, so your practical skills, thinking about control variables, things you keep the same. In this type of question, they do give you a lot of clues. Okay, so first of all, they talk about lactose intolerance. Okay, and then it talks about what the scientists did. Okay, about drinking milk, same volume of milk, and they recorded the symptoms using a scale. Now, immediately I look at that and go, scale, that's a bit subjective, because my 3 might be somebody else's 5, for example. The scientists split the volunteers into 2 groups. Okay, they don't tell you anything about the 2 groups, do they? They don't tell you who they are, their age, their gender, etc. Then it tells you what those 2 groups are. Then it says after 1 week, the scientists changed the type of milk given to the volunteers. Okay, so they want to see the differences, maybe use them as a control. So they're using each person as a control. Okay, so going on to the first question, 5a, says suggest how the scientists may have treated the milk. Well, they would have treated it specifically using lactase. Okay, so they'd add lactase enzyme because that's what's going to break down the lactose. Okay, most of you got the next question correct. It says that they got them to drink the milk first thing in the morning rather than at bedtime. It says suggest why. Well, very basically it would then allow you to record the symptoms during the day whilst you're awake. So if you drink it during the night time, you're not going to be able to record the symptoms. 6c, it says suggest 1 instruction that the scientists would have given the volunteers about what they should not eat or drink each day during this investigation? Well, basically you don't really want to eat any other foods containing lactose or untreated dairy foods or any more milk. Basically, if you mentioned cheese or yogurt, you would get the correct answer. Okay, yet another suggest. So there are plenty of different answers. Again, 1 mark. Why the scientists changed the type of milk they gave after 1 week. Basically, you're eliminating the variation in the volunteers' response to lactose, basically. Or you could say that each person acts as its own control, or to compare the effect of lactose on both groups, which I think is probably a better answer. Okay, on to question 5e. Okay, and it says the volunteers were asked to record 3 symptoms. They used a scoring scale from 0 to 5. 0 is no symptoms, 5 is severe symptoms. Do we know what that means? No, we don't, because there's no more information. And they give some information here about the symptoms. So 3 symptoms: bloating, stomach pain and diarrhea. And then they show drinking untreated milk versus drinking lactose-free milk. And basically, I keep on saying basically, but you can see there's very little difference between the untreated milk and the lactose-free milk. Okay, there's a bit more bloating, but not vast amounts, and a bit more stomach pain. And that's a real key thing there. The differences are so small that if you were to do a statistical test, they wouldn't be significant. That's the first thing I'd be thinking about when comparing the two. That's how my brain would work. So it says, what can you conclude from the scientists' results in table 3? Okay, well, this answer, you could use some of the data, that's fine, but you obviously want to talk about the bloating, you want to talk about the diarrhea and the pain, but that won't get you the full 3 marks. Okay, so the first thing is drinking milk causes bloating. You could say it causes a little bloating, maybe a bit more with untreated milk, and then drinking untreated milk has little or no effect on pain or diarrhea. So again, there's very small effects. You see, if you look at the figures for diarrhea, it's 0.1, whereas it's 0.3 for lactose-free milk. So those would get you the 2 marks. The third mark, though, that many of you missed out on was that the differences are so small. You can see the differences are so small that they may not be significant. So that's with your statistics head on. Okay, you can't judge significance from the fact that there's no real significant difference. Then on to the final question, 5e2. It says suggest. I love the fact suggest is there. Why the scientists' results might be unreliable. Well, the key thing is, and the key thing you need to know is that it is subjective. Having a scale of 0 to 5 is very subjective. One of the things that you can't say is that they were lying. Okay, people don't tend to lie. They tend to tell the truth. So subjective scales, and then you could go on to explain why it is subjective, but if you just said it is subjective, you would get the mark. Here are the answers to question 5. Okay, on to question 6 in your exam pack. This question, explain what is meant by a phylogenetic group, for 1 mark. Well, the answer needs to mention that the group is according to evolutionary links or evolutionary history or relationships or a common ancestry. You can't say things like they're closely related or characteristics that are similar or genetically similar. It really wants you to talk about evolutionary links. Going on to explain what is meant by a species. So again, if you've done your revision, you'll know that for 2 marks, able to reproduce, and they produce fertile offspring. There are plenty of other answers that they will accept, but there are other answers that they won't, so I would look at the mark scheme carefully in a minute. Right, okay, next one, they look at the classifications of tigers and clouded leopards. Okay, and they show similarities and differences in terms of kingdom, order and species. And the question 6b, it says complete table 1 by adding the 4, so you can see the 4 gaps of other taxonomic groups to which the tiger and clouded leopard belong. And as you can see, the answers are class, order, family and genus. Okay, they'll accept the plural answers, phonetic answers as well. Okay, but they have to be in the correct order, just for 1 mark though. Okay, information for the next question is so large it doesn't fit on my screen, so you'll have to look at your exam paper. But it does tell you the circles represent the hierarchy of taxonomic groups for the classification shown in Table 1, and then it says draw additional circles on Figure 1 and label them to include all the information about the tiger and clouded leopard shown in Table 1. So you got 2 marks, so that's basically the additional circles and the labels. So for the correct answer, the 2 circles with 2 inner circles and no overlap is for 1 mark, and then labels are correct. And you can see actually there's different ways of doing it. All right, you could put Panthera and Neophilis all inside that circle there. I'll just point at that circle. So the circles here, Felidae, okay, basically all of that information is going to be inside those circles there for the 2 marks. Or if you look at these, you can get 1 mark, okay, if you put it in this kind of configuration or in this configuration. Okay, ignore underlining capitals. They'll accept P tigris and nebulosa, and they'll accept phonetic spelling. Quite a lot of work for 2 marks. So for 60, okay, they give you some information related to the nucleotide sequence in a gene in populations of tigers living in different parts of the world. Okay, then it says explain. So explain what the information in Table 2 in bold suggests about the phylogenetic relationships. So again, 2 marks is looking for 2 points. Well, to answer this question, you need to obviously know what a phylogenetic relationship is, and the first one to say is there is definitely a relationship between the South China tiger and the Sumatran tiger, as you can see by the nucleotide sequence. So they share what we call a more recent common ancestor because they have an identical nucleotide sequence. That will get you the 2 marks. Okay, all right, so you could draw a labeled hierarchy. Okay, you could also, it would have also accepted the fact that the Siberian tiger is less closely related to both tigers, but then you need to explain why. Here is the answer to question number 6. Okay, on to question number 7. Great little question here. What is meant by species diversity? Well, if you've done your revision, you'll get that, no problem. It's the number of species in a community. They'll also accept number of species in a habitat, area or ecosystem. And there are other things that they will accept, except for, you can't say the number of species in a population, because it doesn't include the fact that it's in a community. Okay. Question 7b is testing the fact that you know an index of diversity, okay, in terms of what is needed, and obviously it's the number of organisms of each species and the total number of organisms or the total number of species. Okay, this question, they give you some information before we get started. So it tells you the effect of sewage entering a river had on the distribution of organisms. So where sewage entered the river, you found a high density but a low index of diversity. So in terms of number of organisms is high, but the number of species is low. So the question is saying suggest. I like that again, suggest. There's lots of reasons why, how sewage entering the river could explain the scientists' findings. So you've got to be careful you don't get carried away here, because it is only 2 marks. So you might want to talk about the fact that sewage depletes oxygen. Okay, so it depletes oxygen, or it can kill certain species. It prevents many species colonizing or remaining in the area. Sewage can be a food source, but only a food source to a few species, because that explains why there is a low index of diversity. Okay, so some species are finding it as a good food source, and so therefore it will only increase the numbers of a few species. Only a few species will survive. So 7D is actually testing your kind of knowledge of your kind of not apparatus and techniques, but how to do science, basically. So it says that a second scientist, so someone different, repeated, that's a key thing, the investigation. They obtained a higher index of diversity. And the question is, explain how the second set of results affected the ability of the scientist to make any conclusions. Well, basically, it's suggesting there that if they've repeated it, the results are not repeatable. They might be unreliable, or they contradict, or they're different, or they don't agree with each other's measurements, and therefore, quite simply, you can't make a conclusion. So it says, how does it affect the ability of the scientists to make any conclusions? Well, they can't make any conclusions, okay, from that experiment. So the next question is then saying, well, suggest how you could make that repeatable, okay, before they can make any conclusions. Well, they only did 2 different tests, okay, so they need to repeat it. They need to do repeats to find a pattern or a distribution, okay. Again, there are lots of acceptable answers that are in the mark scheme coming up in a minute. Here are the answers for question 7. Okay, on to question 8. We're nearly there, not too many questions to go, and there's quite a lot of recall in this. So it's asking you to name the structure. Well, that's recall. It says how gases enter and leave the insect, and that will be the spiracle or spiracles. 4A2, name the small tubes that carry gases directly to and from the cells. So you're going to need either tracheole or the trachea. Okay, so if you said tracheoles, that's fine, but you don't get anything for saying system. Okay, so question 8b, it says explain. All right, so it's explain the movement of oxygen into the gas exchange system of an insect. Well, again, for 3 marks. Okay, so it's looking for quite a substantial answer. All right, the first thing is to say what the oxygen is being used for. So the oxygen is used in respiration. So the way that it moves in is the fact that oxygen, there's a large concentration gradient, okay, from the outside to the inside. So oxygen diffuses in, so you've got to use the word diffuse. You can say oxygen moves down a diffusion gradient. You'll get 2 marks for that, okay. But the key thing there is just to say, well, what is the oxygen going to be used for? Because it doesn't say describe, it does say explain. Okay, so some of you may have missed that mark. So question 8c talks about abdominal abdominal pumping takes place during vigorous activity in insects. Okay, so there's lots of information here about what it is, and the fact that the scientists measured the pressure in the tubes and the volume of carbon dioxide released. Okay, so they give you some nice information there. Again, they don't give you any figures whatsoever, so obviously you're not going to use any data, okay, when you describe your results, but you've got to talk about these peaks, and particularly you notice that the abdominal pumping, and you get a resultant increase in carbon dioxide. So the question is saying describe and explain the results, and again for 3 marks. So the first thing, you get 1 mark for describing. Okay, so we need to describe from using these results, and you can see that the abdominal pumping is linked to the carbon dioxide release. So when the pressure in tubes increases, so does the carbon dioxide released. For the second answer, so you've got to explain it. Well, the pumping raises the pressure in the body, and therefore carbon dioxide can be moved out, so it can be pushed out of the body, or it moves down a pressure gradient. Okay, so it's not straightforward diffusion because the pressure is actually pushing that carbon dioxide out of the body. In terms of questions, that's a nice question, okay, because they give you pretty much most of the answer there. Okay, so that, you know, if you can work out the answer from just looking at that information they've given you. Here are the answers to question 8. Notice there are quite a lot of comments, okay, related to alternative answers, and particularly things that they would reject as well. So look at those carefully, please. Okay, question 9, and it's talking, well, it does tell you that it's talking about mitosis or meiosis or meiosis. So they've got lots of information here, okay, that you need to look at carefully. Okay, so 5A is saying complete table 3 with a tick if the cell shows the feature. So homologous chromosomes can actually be seen in cell B and in cell C. Okay, so woo, they can. Wait a minute. Cell B and cell C. Let's have a look. So if I just, so you can see in cell B you've got homologous chromosomes, in cell C, but they're not. So a homologous chromosome is a pair of chromosomes. Okay, so hopefully you got that correct. And then a stage of mitosis, well, actually the only stage of mitosis there is going to be in cell C. Okay, so if I just put that correctly there, so in cell C. Okay, next question, it says describe and explain the appearance of chromosome K in cell C. So we need to have a look at chromosome K in cell C. Okay, so there's chromosome K, looks pretty much like the other chromosomes. And very basically, it just wants you to talk about the structure of chromosomes. So the fact that they have 2 chromatids connected at a centromere because the DNA is replicated. Or you could say K is on the equator of the spindle, so you can see the equator. So this bit here is the equator of, sorry, that bit there is the equator of the spindle. And it's attached, you can see it's attached at the centromere. So that's okay. 5C, explain what is happening at point J in cell B. So you need to go back, you need to look at point J in cell B. Okay, so if we look at this, so point J, okay, you can actually see, if you look very closely, you can see that the homologous chromosomes are crossing over. That diagram is nearly as good as the diagrams that I produce. But yeah, you can see it crossing over. You could say crossing over or exchange of alleles. Recombination, that's just for one mark. For the second mark, you need to say between the homologous chromosomes. So crossing over between the homologous chromosomes. Okay, for 1 mark next, it says use the information in figure 4 to explain how the number of chromosomes in cell D was produced. Well, if we look at cell D, you can see that there seems to be half the number of chromosomes. So basically what's happened is there's been separation. Okay, so it's looking for separation or segregation of pairs, or the separation of the chromosomes. You could say as a result of meiosis. Okay, you could say that they're pulled to the opposite poles for separation, but basically you can see they are separated. Okay, and 9E, it says it shows the figure 5 shows the mass of DNA present in cells of a population of healthy cells where mitosis is occurring. So it shows the number of cells with this mass of DNA and the mass of DNA in the cell. Okay, so 0, 1, 2. and the number of cells. Now this question, there isn't really much you can say. It only wants 1 mark here, so it doesn't want you describing anything, but you can see some cells contain a mass of DNA between 1 and 2 arbitrary units. Okay, so some have more numbers of cells with this particular mass of DNA, and it's just that the DNA replication is taking place, or you could say that it's not finished. Okay, so the DNA replication is taking place is the answer. Here are the answers to question number 9. Notice again that there are lots of different answers that they will accept, so just double check that, but this is very keyword heavy, crossing over, separation, replication, so quite an interesting question. Okay, on to question number 10 in our mock exam. So it says structure of a leaf. He viewed temporary mounts of leaf tissues with an optical microscope. Right, so fantastic. Describe how temporary mounts are made for 2 marks. So literally you've got to describe, you've got to recall what you may have done in the class. So you obviously need to take a very thin slice or section of the plant, okay, so of the mustard plant leaf. So you take a thin section, you put it on a slide in water, or you can mention in a stain like iodine, and then you add the cover slip. If you said you put it between 2 slides, you would get 1 mark for that. Okay, so the next question, it says calculate the distance between G and H. So you got to measure G and H, okay? Alright, so when you measure it, it could be between several different things, but I'm going to say it was 63 millimeters, okay? Could be 64 actually. But then you've got to measure this distance here. It gives you, it says that distance there is 50 micrometers. So we can then divide it by that, which I measured out as 16 millimeters. Okay, so 16. So that equals 4. Okay, so that's 4 times by 50, because we know that distance there. Okay, it's 4 times 50, which is 200 micrometers. The next question is really testing your math skills, really. So it's saying describe how they could have used temporary mounts of leaves to determine the mean number of chloroplasts in mesophyll cells of a leaf. Well, the first thing you're going to have to do is you're going to have to select a large number of mesophyll cells, first of all. So you can select them at random, but you select the number of cells. Then you've got to count the number of chloroplasts in each cells, and then basically you've got to take the number of chloroplasts that you get, okay, and you divide it by the number of cells, and that will give you the mean number of chloroplasts. Okay, you could mention about cells in a field of view. You could say if you have large numbers of cells, could be the field of view. But you're not going to calculate the mean. You don't have to show how to calculate the mean, because it talks about that in the stem of the question. So it just simply wants you to work that out. Some varied answers for this, okay? It says basically there are different types. It's giving you the answer. It's saying there's different types of cells in a leaf with its own function, okay? And it says, how could you describe a structure such as a leaf? Well, obviously it's not a tissue, because you've got lots of different tissues, okay? It's not basically a tissue, as I said, so it's got lots of different types of cells with their own function. So in fact a leaf is an organ, and definitely not, as some of you typed, which I'll just get here, and I won't embarrass anyone. Somebody said it was an organism. Here's the answers for question number 10. And finally on to question number 11 in your exam. Okay, so first two questions really nice. Okay, so here again, what is the name of a position of a gene on a chromosome? There's only one, there's only one answer, and it's called locus. You could have the plural, loci, as well as the answer. Next one, what is meant by genetic diversity? Again, that's something you need to learn if you don't know already. It's basically the differences in DNA. Okay, so you could say also the number of different alleles or variations in a gene pool. They are not genes, okay? It's related to the gene pool or differences in base sequences of DNA. Okay, for 11C, there is a large amount of information, so you need to read it very carefully. So they're talking about genetic diversity of different types of dogs. They give you mean. It tells you a mean value of 1 shows maximum genetic diversity, 0 shows no genetic diversity. And what's most interesting there is they have done a standard deviation, so you can see the range of the data. Okay. So you can see the mean and the standard deviation. Okay, so question C, 11C says, what do these data show about the differences in genetic diversity between these breeds of dog? Looking at that, 3 marks, that's great. So the first thing to look at is the table of results. So here we've got the Jack Russell Terrier. You can see that basically the Jack Russell Terrier has the greatest genetic diversity, and conversely the Bull Terrier has the least genetic diversity. You could say the Bull Terrier is more inbred as well. The Miniature Terrier and the Airedale are pretty similar, aren't they? Okay, but as you can see, they've got here the standard deviation. All right, so if you were to just discuss those 3 differences, you don't just get the 3 marks for just crediting the list of genetic diversities. You've got to mention the standard deviation. Okay, all right, and in this case you can say that the standard deviations do not overlap. Okay, therefore they are significantly different, but you can say that there's no overlap between them, therefore they are significantly different, or there is a significance between the data. And the final question, it says miniature terriers were first bred from bull terriers in the 19th century, and you need to give 1 explanation for the observed difference in genetic diversity between miniature terriers and bull terriers. So you could talk about either the miniature terrier or the bull terrier. I'm specifically going to talk about the bull terrier because basically the bull terrier, there's probably been a smaller population or a genetic bottleneck, and therefore you've got a reduced number of different alleles in the gene pool. If you want to mention the miniature terriers, you can, and you can say obviously you wouldn't get any extra marks. You've got to say either one or the other because it's only 2 marks. Okay, and you need to say that the miniature terrier breeding has included more outbreeding, so therefore less selection pressure, so that increases the number of different alleles. Okay, you can't use the word genes instead of alleles. You've got to mention increased number of different alleles. All right, it's very important to state that. And here are the answers to the final question, question 11. Please remember to go through all of this with your question paper, noting down the things that you understand, the things you don't understand, and then work on the things that you don't understand. If you are still struggling with some of the answers and need help, then please do speak to your teachers. Okay, thank you very much for listening, and I hope you found it useful. Goodbye until next time.